Okay, we're continuing on our goal to build up protein structure. We're finished now with primary structure, the composition and order of amino acids. We're now done with secondary structure too, the regular repeating patterns of main chain hydrogen bonding. That got us the building blocks of our proteins, alpha helices, which are like columns, and beta sheets, which we use the analogy of like walls or floors if we're trying to build a building. And of course, beta sheets come in parallel interactions and anti-parallel interactions. So at this point, we've got these structural elements, and it's finally time for us to actually go ahead and build the actual protein structure, the three-dimensional full structure. Tertiary structure is what we refer to as the overall fold. It's like we take columns and walls and floors and we finally stitch them together to make an actual building, a three-dimensional functional structure. Now, when we were working on secondary structure, as we said before, we're mainly interested in main chain hydrogen bonding. And we're still interested in that, but we're going to switch our focus more to side chain interactions in tertiary structure. And we'll see how that works out. But remember, the side chains of alpha helices and beta sheets both projected out from those structures, and that's going to allow them to interact with each other. So let's get on to that. Tertiary structure is going to be illustrated by two proteins that we've already talked about before. You may remember in our discussion of sequence alignments that we looked at how the structure of human proteins such as myoglobin and two different forms of hemoglobin can be aligned to each other. And we pointed out that they have similar sequences, and if sequence gives structure, then a similar sequence must mean similar structure. Likewise, a similar structure may give us similar function. So let's take a look at the actual structures of hemoglobin and myoglobin on the next page. I want to introduce these proteins to you in a little more detail. This is going to be myoglobin, and rather than write out the full structure, we're just going to call that MB for myoglobin. And myoglobin is in the muscle. It floats around in the interior of your muscle cells. Hemoglobin, HB, is going to float around in the water, the aqueous environment of your blood. And what we can notice right away, if we look at these tertiary structures, is they have a very similar tertiary structure. So let's get into that by writing tertiary structure here at the top of the page. And we sometimes use 3-0 to describe or to abbreviate tertiary structure. Now, I want to tell you that we're going to talk about a very specific type of tertiary structure. We're going to be discussing what we call globular water-soluble proteins. Let's pick that apart a little bit. Globular refers to proteins that are roughly spherical in shape. Now, these two proteins are actually a little more like eggs, but nonetheless, they're still pretty close to spherical. They're kind of a big blob. Second of all, we're interested in water-soluble proteins. Like I said, both myoglobin and hemoglobin have to float around in the water of either muscles or cells. That means they need to be water-soluble. Now, it turns out tertiary structure is actually a very simple thing for us to describe, especially if we just focus on globular water-soluble proteins. There's really not that much to say. First, we are going to say that you're going to have polar side chains facing out. That, I hope, makes sense. Let's pick an alpha helix here on myoglobin and say that if you're going to have polar residues on that, they would be likely to face outward, interacting into the water environment, which of course is very polar as well. Likewise, we could say that nonpolar side chains are going to face to the interior of the protein. Let's point out that that would be on the other side of this alpha helix. And I hope that makes sense to you. Nonpolar residues don't want to interact with water on the outside. They want to cluster away from water with other nonpolar residues, and they're going to do so because of the very strong driving force of the hydrophobic effect. Now I chose this alpha helix because looking end on, we can see how the side chains radiate out from the main chain core column structure. And you can see now why we might want to paint one side of that column with a particular type of chemical property, nonpolar for instance, and the other side with a different opposite chemical property, in this case polarity. That's how we can generate structures that fit a particular structural purpose in a particular protein. So I also want to say one other thing about these globular water-soluble proteins. Those residues that are facing the interior are tightly packed with each other. So we would say the interior of this protein is tightly packed. And you may say, well, it doesn't look like it. I mean, after all, let's see spaces here and here. But understand, it's the side chains that are going to fill in those spaces and pack up very tight next to each other. There's not any water, very little water on the inside of proteins. Remember, we have 9 nonpolar amino acids. Some are big, like tryptophan and phenylalanine. Some are small, like lysine and alanine. We got all sorts of different shapes, like isoleucine and valine and methionine and things like that. We can use those like jigsaw puzzle pieces to fit together and form a nice, solid, packed interior that makes the overall protein very solid. So that's really it about tertiary structure of globular water-soluble proteins. Let's just point out that if we have similar sequences, like we said for these two proteins, then you would expect to have similar but non-identical protein structures for them as well. Take a look at this. We have a beautiful helix kind of going north-south here. We've got something very similar here. We got one going off to the northeast away from us in myoglobin here, and one that's taking the same orientation here. We've got that helix we're looking to end on here. We're looking at the same end on helix right there. So similar structure, but not identical. And we're going to learn in the next chapter that they're similar function, but not identical. So that's really it for tertiary structure, at least for globular water-soluble proteins. We're going to look at these proteins and their function later in the next chapter. But before we do that, we got to finish this chapter with moving on to quaternary structure. One level of protein complexity left.